network of protein fibers in the cell. Makes a scaffold or connects at the surface. Some parts of the cytoskeleton connect to cell adhesion molecules to give a strong connection to the other cell instead of, you can't just pluck the protein out of the membrane because it's attached to all these other things inside. In fact, I'll go ahead and add them here. Organelles in the cytoplasm. The nucleus is also considered an organelle, but it's not part of the cytoplasm, it's its own thing. This is the list. Ribosomes, endoplasmic reticulum, which comes in rough and smooth, vesicles, Golgi apparatus, mitochondria, lysosomes, and peroxisomes. We'll look at the functions of each of them, so you don't have to worry about getting them all written down now. Ribosomes, this means ribose body, ribose as in ribonucleic acid. They're made of RNA and protein, and they are these little globs. They'll show up on the electron microscope as dots. Their job is to build proteins. Some of them are what's called free ribosomes, where they're just in the cytoplasm, and they'll make proteins that stay inside the cell. There's also some called fixed ribosomes that are on our next organelle, the rough endoplasmic reticulum. Reticulum means little net. Endoplasmic means inside the cytoplasm. The endoplasmic reticulum is folds and tubes.
What did you say the word meant?
Little net inside the cytoplasm. So some of it is flat stretches like folded up fabric. Some of it's more like coral tubes, and when you take a cross section through it, you'll see a bunch of irregular shapes like this. The endoplasmic reticulum builds and transports substances in the cell. The rough ER has ribosomes on it, and that's why it's called rough. The rough ER is going to build proteins, and the ones it builds, as opposed to the free ribosomes, are proteins that need to either be attached to a membrane, because it can build it within the membrane here, or that need to be sent out of the cell. The smooth ER makes lipids such as cholesterol or phospholipids to repair the cell membrane, and it also detoxifies some drugs. So here's a semi 3D representation of them. So here where we've got these openings, it's like sliced through. And that's how you'll usually see it, is as slices. So the rough ER is flatter folds, kind of long and thin, and has ribosomes all over it. The smooth ER is more like rounded tubes. When we cut it like this, it looks kind of like coral. Dumb joke that I got from the Ball State Theater Department. What did the smooth ER say to the rough ER? Is that a ribosome on you, or you're just happy to see me? Questions on endoplasmic reticulum? Vesicles are little bubbles used to transport substances. Some of them will come off of the endoplasmic reticulum. They can come from the cell membrane. Sometimes the cell will take things in and pinch it off into a vesicle. The third place they come from is the next organelle, the Golgi apparatus. Let's see, I'll put it over here. Golgi was an Italian guy around the turn of the 20th century who studied a lot of things in anatomy. And there used to be a bunch of other things named after him too. Most of those we've switched over, but we've kept this one. The Golgi apparatus, shown in purple here, after the rough ER makes a protein, it puts it in a vesicle and sends it to the Golgi apparatus. And then the Golgi apparatus processes that protein. That could include making some changes to it, like cutting out a piece or attaching carbohydrates or some other molecule to them. And it sends them where they need to go. So it repackages them in a new vesicle that basically has an address on it that says this is where this needs to go. That would be some kind of surface chemical. So this shows one being sent to the cell membrane to secrete that protein. I like to call the Golgi apparatus the mailroom of the cell because it designates where everything's going to go. And it kind of looks like a cross between rough and smooth ER because it's flat like rough, but it doesn't have ribosomes. And plus it's more compact in one place instead of spread way out. Mitochondria, I'll just label the one I've already got. Their job is cellular respiration, getting energy from your food. And that's another thing we'll talk about, how it works in chapter 4. This structure with two membranes is important. The outer membrane forms the shape of the mitochondrion. Singular ends in ON instead of A. I'm not going to be too particular about that, but you'll run across it, so that's what you're seeing. The outer one gives it the shape, which is usually kind of bean-shaped. It might look round if you slice through it the other way on the slide. The inner membrane is all folded up. The little folds are called cristae, which means crests. The purpose of those is to have lots of surface area for chemical reactions. There's a bunch of enzymes on the inner membrane that do the reactions to get the energy from the food. The space inside the inner membrane is called the matrix. Lysosomes look a lot like vesicles. On an electron microscope, you can kind of tell the difference because they're clumped up with stuff. Vesicles, you often can't see what's in them. But they're just bubbles with proteins in them. But instead of carrying that protein somewhere to deliver it, it keeps that protein in it. Those proteins are digestive enzymes. So lysosomes are kind of like the stomach of the cell in organ terms, or like a recycling center. What they do is break down things that you don't want to keep intact in the cell, which can be old organelles that are wearing out. It can be something that's been taken in, like if a white blood cell eats a bacteria. They also break down some drugs and toxins like the smoothie R does, especially in the liver. Here's a lysosome animation that includes how they get built, which comes from the rough ER and the Golgi, and shows them digesting things.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles, such as old mitochondria. This results in the destruction and recycling of these structures.
So there's a couple of extra terms on there. If it's not on your vocabulary list on the study guide, don't worry about it. But overall, that's how your lysosomes work. Questions on lysosomes or mitochondria?
Lysosome goes through the whole cell, or just through the things that just showed, goes throughout the whole and taking out all the bad, right?
Yeah, there's going to be multiples scattered around the cell, and when something needs to be broken down, they go there and break it down.
Okay.
The last one is peroxisomes. These are like little isolation chambers, like the hoods in the chemistry lab, where you don't want to do it out on your bench and have nasty fumes coming out for you to inhale. There are some chemical reactions where they take a couple of steps and somewhere in the middle is something nasty that you don't want loose in the cell. So you do it in the peroxisome. The reason we call them peroxisome is one of the intermediates is peroxide, depending on the reaction. So the peroxisomes do the reactions and they break down whatever is hazardous, or they change it in a way to make it not hazardous. Some things that they're used for are producing bile, because that has some hazardous intermediates, and breaking down alcohol and some lipids. The some, again, is body. So these are peroxide bodies. Lysis means breaking down. So lysosome is a breaking down body. So, cytoskeleton also gives us some organelles, including a centrosome, which is one on the inside, cilia and flagella, which are at the surface and used for movement. And then these other things here, microfilaments, microtubules, and intermediate filaments, are the structures in this network of proteins. The centrosome, also called centrioles, is a pair of cylinders near the nucleus. They're always at right angles to each other. Centrosome is center body because it's near the nucleus. In a more detailed view, it's basically a bunch of sticks that make the cylinder. Those are microtubules, like in the cytoskeleton in general. Centrosomes are used to move the chromosomes, which is the pieces of DNA, during cell division. And they build other parts of the cytoskeleton, including cilia and flagella, which are our next ones. Cilia means eyelashes. Cilia make a whole surface of little tiny hairs, and they move to push things along the surface of the cell. There are some single-celled organisms that use cilia to move their whole cells. In our bodies, we have cells that use cilia to move things along their surface, like to clear out dirty mucus from the respiratory tract. Flagella, singular is flagellum, because a cell that has it will just have one. It means whip. A flagellum is a long tail that is used for swimming. We only have one cell type that swims, and that's sperm. Questions on cilia, flagella, centrosomes?